Hello, I'm Neil Spooner, the Editor-in-Chief of Bioanalysis. I'd like to welcome you to today's Bioanalysis Zone panel discussion. We're going to be focusing on bioanalysis of oligonucleotides, and I'd like to welcome all of our panel to the discussion today.
Hello, I'm Seamus Lam. I'm the Senior VP for Bioanalysis in QPS.
My name is Awa Liu. I'm the Executive Director at Resolium.
Hello, my name is Dominic Guarino. I'm the Senior Director of Scientific Services for KCAS Bio.
Hello, my name is Luca Ferrari. I'm Strategy Area Leader for Small Molecule Bioanalytics and Biomarkers at Roche.
So welcome all. I'm really looking forward to this discussion today, focusing on oligonucleotides. I think they've become an important part of the pharmaceutical industry and therefore an important consideration for us as bioanalysts to figure out what to do with them and what we should be doing. Dominic, I know you volunteered to give a little bit of background to the importance of oligonucleotides and why we need to measure them.
Yeah, when you think of oligos, they're not new per se. I think they're all the rage nowadays, in part because of some of the advancements in the technology, but people have been treated with oligos for a number of decades now. They were more like ASOs and morpholinos kind of back in the day, and they were in general treated for like some rare diseases and other applications. But if you flash forward to today, there's a lot of antisense oligos, a lot being treated for a wide range of things. They're also being attached to like proteins, the ARCs they call them, antibody siRNA conjugates. And so in general, the field has really advanced quite a bit in oligos, and with that comes some bioanalytical challenges. And so I think in the past you had maybe not had as much demand for it as you do today. And I think that's led to this panel discussion here. There's just quite a lot of demand for oligos. And as with any sort of bioanalytical thing, everybody's looking for better sensitivity and more robust assays, and we'll dive into more about how we go about doing that. But I don't know if my colleagues want to add anything to that, but in general, that's how I see oligos. They're not new. I know they're very safe, but when you start putting them onto payloads and things like that, then there is some efficacy and safety concerns. But in general, oligos are powerful and certainly not going anywhere in terms of their therapeutic, future.
Absolutely. And do you notice how big a part of your portfolios they are? How much of your workload is oligonucleotides, would you say?
In my division, it's about 40% of oligos using mass spec. So it's quite incredible from that perspective. I mean, I think in about 4 years, we grew about 4 times the amount of oligo work we have done by mass spec.
Wow, that's a lot. It
is a lot. I think it's really is, as Dom says, right, is no longer just a single strand ASO. Is now double-stranded, and then all these conjugations that just change the whole chemistry and the thinking process.
And what kind of bioanalytical challenges does that bring?
So I can start from that perspective. So historically, the first generation to the 4th generation, there is some stability concern. Nowadays, the 4th generation, they're pretty stable. The issue is that despite we feel there is some stability, there is actually some metabolism. And the metabolism is normally through exonucleases, right? So you actually lose 1 mer and 2 mer and so on and so forth. So the methodology, you actually have to separate a 20 mer versus a 19 mer versus 18 mers. And then the sensitivity is very important. So it's not just a separation. Sensitivity is actually understanding technology involved.
Okay. So, and you talked about metabolism there. Are the metabolites biologically active as well? Do they need to be measured as well?
Typically focus on the N-1, N-2, or even a little bit shorter of the metabolites. So as Zema said, it depends on what kind of technology we use for that type of information. At the early stage of the drug development, we always want to understand the metabolites. So at that time, we can use the high-resolution mass spec for that.
Okay. So what kind of technologies above and beyond high-resolution mass spectrometry are you using for measuring oligonucleotides?
Well, I can tell you, depending on because oligonucleotides, we may have different types. So we spoke about antisense oligonucleotides, we spoke about small interfering RNAs, which are double-stranded. We have important chemistry modifications which have been introduced in the backbone, and we have conjugated ones, right? So these are the kind of oligonucleotides we are mainly playing with. Technology. I cannot say that we have a first-line technology. It depends on the kind of oligonucleotide that we have and the intended use of the assay. So, and also our strategy, I mean, evolved as long as the technology also evolved. So I can tell you that 10 years ago, it was very difficult to use mass spec because the sensitivity was a main constraint. Now the situation is very much different. So we have different understanding of the use of the technology, and we can adapt that based on what we need to measure.
Yeah, maybe I'll add a little bit, like, so you can still measure them by PCR, right? That's an option. There's the mass spec option, and then there's the hybrid ELISA option. And, you know, we were kind of talking earlier, and I said, oh, you should do them all by mass spec, and that was too big of a generalization. That's just, I think all it takes is one experience with something, and say, oh, we should try it that way, but we can dive in more around that. But it is, as my colleague said, it is definitely something. You look at the biology, you look at the drug itself, and then also, you know, some of it has to be what do you have available too. I think that is an important factor when you go to look at how you measure them.
Yeah. If you look at the FDA-approved drug, as of 2023, there were 20 oligonucleotide drugs approved, and including 13 azoles, 6 SN, and 1 aptamer. And then they also listed the technology used for all these drugs. At the early stage, I think back to 95, at that time, used the kind of LCFD or LBA technology. But in the most current 10 years, I think LCMS has become more and more dominant technology to measure the oligo bioanalysis so far. But if we need the sensitivity, we still sometimes have to go back to the LBA. So the LBA and the LCMS technology, they have their own pros and cons. The LBA has higher sensitivity, but the LBA cannot differentiate the parent and the metabolite. So that's why their specificity or selectivity is not that good. And the LCMS, the sensitivity is not as good as the LBA, but LCMS has the LC part and also mass spectrometry part. Both of them can provide very good selectivity. So that's why LCMS technology has good selectivity and specificity. But LBA is more is faster because high throughput. The data reading only takes 1 minute for one plate. LCMS, you need to inject one sample 5 minutes, 6 minutes. So one plate will take 600 minutes in total. So they have their pros and cons.
There are various parameters which have to be considered, in my opinion, and also are we talking about application in the preclinical stage? Are we talking about clinical applications? Because in my opinion, something that we have to consider is the fact that LCMS is reagent-free. And a ligand-binding assay like hybridization ELISA is not reagent-free. And so when it comes to supporting discovery projects or in the preclinical stage, we prefer to be fast and use LCMS as much as we can, and then maybe adjust the strategy based on the way the project develops and what the needs are. But I fully agree with what you stated.
Yeah, and it can be a challenge sometimes to find an antibody that can bind to that piece of real estate uniquely, right? There's a lot of promiscuity with those antibodies, so there is an advantage there with the LCMS not needing that. But, you know, if you can find an antibody that binds with high affinity to your oligo, then you are going to be able to have a more sensitive LBA method putatively, right? It all has to be tested. But
well, more typically, we would use hybridization ELISA for that. So you use a capture and a detection probe, and these bind to the analyte by using a hybridization mechanism. So
yeah. So when I say
don't
LBA,
use antibodies, but yeah, sure.
When I say LBA, I mean hybrid ELISA. But he is absolutely right. You can't. It has to be a hybrid ELISA format to do it.
So the LBA platform for the oligonucleotide is slightly different from the biologicals. So for the biological, for the capture region and detection region, we usually use the affinity by antibody, you know, this approach. But for the oligonucleotides LBA approach, like the ELISA or MSD or even JALOS, actually we design a probe. We can use either a single probe or single probe or double probe, and then can utilize that one to capture. And then the detector region could be, if the ELISA you use the colorimetric, if the MSD you use the fluorescence detection. So that would be the difference.
Also, you go the other way around, because typically we tend to use in research and early development LCMS, so we can get information about, typically the sensitivity is not a challenge at that stage. We try to get as much information as possible regarding the biotransformation products, and then based on that, we may decide which technology to use.
That's for the high resolution mass spec.
Yeah.
Right, I 100% agree. I mean, I think for the early development, getting as much information as possible, and you use some type of mass spec, you actually get that, right? So if you use a hybridization, you're actually pooling your parents and your N-1, N-2, and so on and so forth, and you detect it. So all you have measuring is actually what your hybrid probe pulls down, right? So there's no metabolized information other than a whole jumble. So by using mass spec, whether you're using high-res, you're using triples, you get some understanding of potential metabolites there are, right? And I think sort of looking at the structure itself is also give you some comfort in reality, because if you have A at your 5' end or your 3' end, normally there is oxidative deamination, right? So you can't really analyze that other than by mass spec. And actually it's a little bit difficult using triple because it's only 1 AMU. So using high-res is more less problematic, right? So you think that, hey, I'm going to use high-res mass spec for everything. And then with an LC in front, everything is simple, right? It's actually not as simple as we all like to think, because nothing is that simple. Because every time you have a phosphorothioate, you actually generate diastereomers. So you have 2 phosphorothioate, now you have 2 to the power of 2, you have 2 to the power of 3. And you would think that, hey, it's okay, they won't separate on the column. They actually do separate on the column. And they separate in column very tightly, so you have a retention time of probably 10 seconds. And then you have your N-1 on top of that. And then you have your oxidative deamination on top of that. So having a very early read using high-res is very important. And once you know there's no metabolism, use the cheapest, quickest method. That is the most important thing, right? So, yeah.
And so what kind of chromatographic approaches are you using to do these separations early on when you don't know much about the metabolism?
So we use ion pairing. I don't really like ion pairing, but that's the only way that actually you get what you need to get done. Because we do so much, we actually have 4 high-res mass spec with ion pairing, just 100% for those. We have tried to use Helix. Helix is nice, but it doesn't get enough sensitivity. So you get about 2 orders of differences from that perspective, right? So, and the hybridization ELISA is wonderful because if you do it right, you can get about 10 picograms per mil for a single strand. Double strand, you also only measure 1 single strand, right? So that's a problem. If you're using a double-strand siRNA using high-res, you can actually measure both strands, so that understanding of like potential PK, that's helpful. The problem is that a single strand using a traditional mass spec, traditional LC, gives you 1 nanograms. So 10 picograms, 1 nanograms is a lot different, right?
2 orders of magnitude out there.
Yeah. So, but if you now with a new mass spec, whichever flavor of a new mass spec you want, with micro LCs, you can actually go down now to 50 picograms. So it actually is incredibly helpful from that perspective.
So you are bridging that gap.
There is definitely the gap is getting narrower and narrower. My next favorite topic in the future, in about 2 years, is actually trying to put all this stuff in the SFC.
Okay.
Because then you get a huge amount of chromatographic resolution. But in theory, it should work, but we won't even know until we get 2 years of data.
And it might be finally a use for SFC, which has been hanging around there all my career.
Yep, so did that at postdoc, and I'm sort of determined to go back and do it useful.
No, it's interesting. So where are the high-resolution mass spectrometers now in sensitivity versus what we're used to with triple quads for small molecules with selective reaction monitoring? You said we're down to 50 picogram per mil.
Yeah, so if you put a micro LCs in front of that, you put a decent mass spec, you got the 50 picograms. The important part, right? We will only use high res when we know there is metabolism. If there's no metabolism or very low metabolism, we use a triple. If we know there is irrelevant number of metabolites, then we just go use hybridization ELISA, right?
Okay.
I think the most important part is you must have all the tools, choose the right tools to solve the right problem.
I have a similar approach. I would say we don't use high resolution since the beginning. So we typically start with triple quads. But I mean, in our company, we use high resolution to elucidate and to provide information on the metabolites of the oligonucleotide drugs. And then based on that, we make decisions how best to proceed. Something which I wanted to highlight in terms of sensitivity, we evaluated high resolution mass spec in the past, also for the quantitative analysis. So when we know exactly what we need to determine, because one of the concerns we had at the beginning was that our LCMS assays on triple quads would suffer from specificity issues or selectivity issues, because in most of the cases, the fragmentation of the oligonucleotides is not so diagnostic, right? So we tend to monitor always a common fragment, and we suspected it would raise concerns about the selectivity. That was not the case, and that is mainly due to the fact that the chromatography that we use must have enough separative power, right? So the efficiency and of our separations and is enough, so we don't suffer from that. I also must admit that the high resolution mass specs available 5 years ago had a different sensitivity compared to, so whenever we try to compare the sensitivity of a triple quad against a high resolution one, I mean, there was no really, it was not possible to compare. Yes. So the high resolution mass specs suffered from a sensitivity loss, which was difficult to compensate. So much higher sensitivity on the triple quads. We use as much as we can triple quads, but we need to have the biotransformation data at hand.
Exactly. No biotransformation, you don't know what you're doing. Yeah. So the other thing sort of interesting, I think where the mass spec is concerned versus sort of like LBA, right? So, and also high-res full scan versus a MSMS, where they're using triples, using high-res to do MSMS, is we're actually running a full scan. If you inject a 10 nanograms per mil, 1 nanograms per mil, whether you're looking at N-1, N-2, N-3, you actually get very, very similar peak height and area count, okay? I mean, that's our experience. I'm not quite sure about what your experience is. When you do MSMS, it's different, but using full scan is very similar. So, and that's the reason why I like to use high-res for that reason, because don't necessarily have to N-1 or N-2. would have a quick estimate of what my potential AUC is of the metabolites, and that gives some comfort to the metabolism folks and the tox folks.
Is it the throughput suffers a little bit? Is there more cost in the discovery setting for that high-res mass spec, or it's just, I understand the biotransformation portion of it, but it seems like you're taking like a sledgehammer to...
So this is why I always says, if you know metabolism, don't use high-res mass spec, because when you run 3 plates, 96 well, you flow on a triple MSMS, maybe 2 megabyte data, ligand binding hardly any. A 3 plate on the high res is 3 gigabytes. So so you have to pick your tools to do what you need to do.
I'm interested about the chromatography bit, because it sounds like the capillary LC separation is an important part of this getting the sensitivity. So are you finding the capillary LC separations are robust and reproducible enough? Because that wasn't always the case with capillary separations.
That is true. I can tell you my experience. So we started and we are still using high flow rate separations typically on UPLC columns. But there is an important thing that we have to consider for the oligonucleotides. So we need to reduce the dimension in internal diameter of our columns, because so we don't have that much biological matrix to play with. So depending on some therapeutic areas and the animal model, if you're talking about the preclinical, in some cases we can have just 2 or 3 milligrams of biological matrix to play with. So the sensitivity needed is related to the miniaturization of the samples that we have, right? It's a true need. So I believe that the microflow chromatography can be considered as robust as the high flow rate chromatography now. So we have much better equipment. And what we are trying to investigate now is whether nanoflow can be also considered, because there is novel equipment, and nanoflow was never considered because of the very long run times. They are not compatible with the PK assay, right? But I mean, we have much better options which can be considered at this moment in time.
Yeah, high flow is about, you can get a injection about 5 minutes. A microflow, we can get about 6-7 minutes, and it gives you a lot of options. We haven't tried nanoflow. I'm a little bit concerned because the column is not that robust. And if you think of the way that you do your typical extraction, there's a lot of stuff on that thing, right? So flowing in a nano actually is a little bit concerning, but micro, not an issue.
Now we have the nanoflow at lab, but one of the issues we run into is the maintenance of the nanoflow. So it's very easy to be blocked and have the high pressure issue. Yeah, so other than the LCMS and LBA, actually there's another approach kind of emergent probably 2 years ago. It's kind of hybridized LCMS. So they use the LBA sample cleanup procedure and then coupled with the LCMS. That one also is very helpful to improve the sensitivity. But in our lab, we are doing the same thing. So we use the high resolution mass spec to identify the potential metabolite as much as possible, so that we understand we need to separate them. But when we really quantify the samples, we like to use the triple core mass spec, because we did a side-by-side comparison, I think 2 years ago, between the high resolution mass spec and the triple core mass spec. At that time, it's kind of Explorers 240 compared to the 6500 plus. And then we got the similar sensitivity actually. We were interested about the difference between these two types of the instrument. And then we got the similar sensitivity, but the triple core mass spec is more precise. The data is more robust. That's the conclusion we got. So that's why in our lab, we always utilize the high resolution mass spec to understand the samples first, to separate the metabolite as much as possible, and then we move to the triple core for the later quantitative stage.
There is another reason. It's a practical reason, but the chromatography for oligonucleotides is not straightforward as it would be for small molecule, right? So you mentioned we have to use ion pairing chromatography, and at least in our company we have dedicated systems for oligo analysis, which means at least we need to have a dedicated LC system. The mass spec then, but what we are trying to do is to have a CMS rig, so systems which are fully dedicated for the oligonucleotides, and we have plenty of triple quads, and the access to high resolution is more limited, right? So in order to have a critical mass and stuff, and also for a practical reason, we prefer to have the quantitative methods running on the triple quads, fully dedicated for oligoanalysis.
Another issue, as Zema said, the high resolution mass spec, the data file is huge. It's very big, right? So because it's a full scan, the advantage is you can come back to mining the data again to find the potential, metabolites. So that's why in our company, we have more triple quad mass spec compared to the high resolution mass spec method. And for the chromatography part, actually, our parent is one part, but actually oligo itself, they have the PMO tab. PMO is very different. PMO is kind of not the negative charged. The other non-PMO oligos, they're negative charged, so that's why we need to use the ion pairing. But for the PMO or PPMO or other conjugate of the PMO, just the reverse phase is fine. We don't need a dedicated instrument, just the regular can share with the others, yeah. And then sometimes we need to shift from the ion pairing system to non-ion pairing system. We have a procedure right now. Previously, we need to wash the system probably a whole day or 2 days. But right now, we have a procedure. We can do it within 1 day. Couple hours, we can switch it. But we still don't want to do it because it's not efficient. So it's end up having the dedicated instrument for the ion pairing instrument, yeah.
Yeah, so for us is that we have 4 TOFs dedicated for oligos. We have 2 triples dedicated for oligos. The morpholino behaves like proteins, peptides, so they goes into the triples. They also have the metabolism was a little bit different. So there's definitely a difference between phosphorylate and morpholino. So it's going to be interesting with the way that people are thinking about it, right? So so far what we talked about is single-stranded, double-stranded with some type of conjugate, right? And the conjugate can be a lipid conjugate, can be a GalNAc conjugate. So they still behave similarly, right? But you actually start putting a antisense or sRNA, which is a lot of negative charge, like a rod, and then tagged to an antibody, which is positively charged, big, a rod. So the behavior is very different when you're actually analyzing it. So you think that, hey, when you do that, you may want to think the ADC type of bioanalysis it doesn't really work like that. It's really going to be driven by whether you have a antibody, you have a protein, and then a size of the protein, because the physical chemical property of something is usually negative. Something that is not negative, more positive, it changes your chromatography, it changes extraction technique, changes your whole thinking process.
I think that takes us to me what is an important point of all this. It's not just the technologies that you have to go for here, it's the skills of your staff, and I mean that must be quite a challenge as well. I don't know whether anyone wants to talk about that at all.
Well, high-res mass spec is new to KCAS Bio, so I'm gonna have to pause there. As for all these challenges with triple quads, we certainly encounter them. As for staffing, you're right, that is something that requires a lot of kind of skill around the ion pairing. You got to blast it with negative charges to kind of get it onto the mass spec. There are all sorts of different sorts of columns you need to use, right? And then there is the reduction and what is the other alkylation of it, I guess. That has to be somewhat of an art. So for the mass spec side of things, I think there is probably more skilled. It's a more robust platform. It's been around much longer. If you shift over to like the hybridolizes and the ligand binding assay space, yeah, that is something that certainly is a challenge there as well. I think these are, I keep hearing my colleagues talk about picogram sensitivity levels, and you alluded to it briefly when you said, if you can find the right, you kind of said that early on. I do think it is something where Oftentimes you'll get someone that can get a hybrid ELISA to work, but it's hard to train a second analyst. Sometimes that can be tricky. And then oftentimes, we have like a production team or we have a development team that leads into a production team. And oftentimes that scientist will have to follow that method for a hybrid ELISA. I do think like we've seen some of these methods kind of poop out is the word that comes into mind, when if someone leaves the organization, if there's another pair of hands on it, it can be hard to get that sensitivity back. So I think that's a constant challenge with not just oligos. It's a constant challenge to have any sort of talent when it comes to running bioanalysis. But I think it's something that every company is struggling with a little bit. But I do think the one thing that is improving the technology itself is there are more commercially available reagents that are bigger and better and better. And I think that's really helped the whole, making it more robust. If you went back 5 years ago and tried to do some of these methods, you wouldn't be able to find as many maybe potential commercially available reagents, where it's coming to the forefront, some of the providers of them are making better reagents as well. So I think that's been a nice advancement. And I would continue to see that grow, right? If you think of the history of like antibodies against like payloads, for instance, if you tried to buy that 10 years ago, it would have cost you a fortune and they weren't good. Flash forward to today, they're much better. So I think this is an evolution around the oligo space as well. I would expect you'll see more readily available commercial reagents, which I think is probably, the analysts are part of the issue, but you're only as good as what you start with. So I think that can actually help move the field over time.
And also there is another thing which we have considered the LCMS part now when it comes to LCMS bioanalysis. We have to consider also that the extraction of the oligonucleotides is not as straightforward. So simple protein precipitation wouldn't work.
Yeah.
So we have different options here. We can go for liquid extraction. We can do ion pairing, reversed phase SPE. We can do anion exchange SPE. We can use a combination of them. And then we know that we have these tools, but in terms of method development, and we have to think about the nature of the oligonucleotide that we have, the conjugation that we have, how much it is changing the lipophilicity of the construct that we have in our hands. And so in terms of the experience of the bioanalysts involved in the method development, I would say that an upper level is needed.
It took us 4 years to build 18 people dedicated oligo teams that can run high-res. And I was thinking that it would take us 6-7 years. So I was a little bit surprised. I'm thinking back all the high-res I worked on when I was a grad student, right? They next to impossible work. Nowadays, the high-res are pretty easy, right? The softwares they're okay, they're much more robust. So the instrument side, actually, the vendors has really helped out. The chemistry side is definitely problematic, that we need to think a little bit more how to do that. And then the other thing that part of me like hybridization, ligand binding, is because I don't have to deal with stable label, right? I don't have to deal with IS. So with oligos, historically, I is always analog, right? Because nobody wants to build a stable label oligos. But in reality, it's not that difficult, because you just sort of stable label your little monomers, right? Mm-hmm. And then you just put it into a nucleic acid machine and stitch it up. So you can do the same thing whether you're using C13, N15, right, or S34. So you can do all that stuff. It's just get a little bit pricey versus another analog.
Yeah, it depends on the chemical modifications, but I fully agree that also the use of sulfur 34, as long as you have sufficient number of phosphorothioates in the backbone, mm-hmm. can be very convenient in getting stabilized up-labeled molecules. Yeah.
Regarding the talents, I definitely agree with Luca and Zemas. The people working on the oligonucleotides needs extended training for that, right? Couple reasons. So as Zemas said, the internal standard, typically we want to use the stable label internal standard, but currently oligonucleotides will still use the analog internal standard. And the sample has the matrix effect. PPE doesn't work. Have to use LLE SPE or LLE plus SPE. So the matrix effect is another thing. And then the M-parent system itself needs equilibration, right? And the reagent could be expired sooner than the other non-M-parent reagent as well. So all this kind of thing adding together, if we don't understand the oligo method itself, you probably will be very frustrated because you cannot get it work. Just sometimes no peak, or the peak shape is very bad, and the column could be killed very quickly. Some of the tissue assay will end up have to use the LOE plus as a PE. That's the whole day process. That's why our lab recently kind of work on the hybridization LCMS, which can save the column and the sensitivity increase as well.
But I think you touched base on a very important point, which is a peculiar aspect for the oligonucleotides. So if I compare oligonucleotides with other small molecule modalities, I would say that in 100% of the cases of the oligonucleotides, we have to analyze tissues. It's very important to analyze tissues because it's very important to correlate the concentration at the target tissue, site of efficacy, with the key biomarker or target engagement endpoints. So typically for an antisense oligonucleotide, we would measure the concentration of the oligo, we would measure the concentration of the mRNA to check as an indicator of the target engagement. So we analyze the tissue. Sometimes the PD is even more important. So as a result, the amount of tissue samples that we analyze is very high compared to other modalities. And then having a method which is not prone to matrix effects and stuff is absolutely important.
Yeah, so the amount of money that we spend on tissues is absolutely insane. Yeah, it's absolutely insane. So I think we analyzed one time 15 tissues as part of the early development programs, right? Which is very unusual compared to everything else. But I think, and the other thing with analyzing tissues, right, you're always worrying about does your biopsy actually represent what is really going on, right? So as a metabolism chemist or metabolism person, you think of, okay, liver is a well-stirred model, but we all know that it's not factual, right? So you're gonna have to take the whole organ, and then how do you actually make sure that your whole organ is homogenized to go down to the next level? So that's a little bit different, okay? But I think the really cool things about the whole oligos, as Dom says, that in the bad old days, it was really rare disease, and you don't have that many samples. But now the PCSK9, right? So you're addressing all the major disease, and biomarkers is very important because they address what it needs to get done. So you don't have to deal with many clinical trials with many patients, figure out it's going to work or not. So your initial screening of the patients by genomic, whatever you can do it, that will give you what your options need to be, right? So if you are looking at your downstream processes, small metabolism, okay, so you're looking at, or in reality, metabolomics, you can actually sort of tie in your oligos, PK, and then up and down of your small acid metabolites, right? But then if you think of what people are now using oligo address, which is sort of a lot of the muscular disease. So the agency asked for Western, they asked for mass spec, but that data is much more interesting. So doing the PK itself, and then understanding mRNA and downstream all the way down to the protein, I think that is going to be the next step that we as bioanalytical people really have to understand. So for me is when I work on an oligo program, right, I don't think of I'm just working oligo, I'm thinking I'm doing well oligos, I'm going to be doing mRNAs, I'm going to be doing proteins, I'm going to do a small molecule. So it gives you a whole stream of like exercising your brain that you don't used to do unless you're in grad school, right? Because you're trying to learn everything. So it's a really interesting topic from that perspective.
Does that, I mean, having all these considerations and these more complex methods, what are the lead times kind of like for your method development validation? You know, it's sounding like you've got some quite big lead times here, because deciding what to measure, how to measure it, and a lot more complex methods.
I think lead time depends on our resource, but turnaround time could be very different. Yeah, for the LBA, the method development validation may take a little bit longer time, but when you do the sample analysis, it's much quicker. Yeah, but for LCMS, method development validation could be shorter, but when you really run the samples, right, 96 samples, how long you need to spend on it? I think that's the lead time itself. It depends on our resource, how many projects we have. But Zemus mentioned about the tissue stuff and also the knockdown of the protein and this kind of thing. I think we should talk about another technology, which is the RT-qPCR, because for the risk concentration measure, actually we need to use the immunocapture to capture that risk out, and then we usually use the RT-qPCR to measure the concentration, because concentration is very low. So that's another technology we are using more and more to understand the efficacy. Understand if that the drug itself, the oligo, really go to your target and really convert to your target, protein. So that's something also we are working on as well.
Yeah. We're talking about biodistribution here, right? So it's like on target, off target. You said earlier the expense for this is outrageous, especially 5-6 years ago. They were asking you to validate all those methods, right? So now there has been some changes around the regulatory requirements where it's okay to use a qualified method, or even like use a hard tissue and a soft tissue kind of way to go about it. But it still is, it just piles up like 23 different tissues and so forth. And we actually, when we do biodistribution, sometimes it's by LCMS, but we are using more and more qPCR or RT-PCR, as you said. And so getting back to lead times, that has a kind of an impact. Lead times are pretty easy to manage. It's the turnaround time of the samples that becomes challenging. If someone drops 5,000 samples on your doorstep, or 2,000 samples is kind of the size. And so for us, we got a 60-person mass spec team and a 10-person PCR team. So that's going to have a big impact on actually it's an 85-person mass spec team now and about 10 or 12 in our PCR. So that's where it might be more of a challenge with timing of everything. But for us, lead times are always between maybe they exceed 4 to 6 weeks in average in this space for us. And we work pretty hard to drive that down, though. We have some things we do for that.
So in the discovery space or the early development, but still in the preclinical arena, I think it's important that we are fast. So spending extra time in developing an HLISA, we need to design the capture detection probe and stuff for a compound which can be short-lived. And also for the oligonucleotides, I think that in particular for the ASOs, a big attrition happened in the preclinical, so in the toxicological studies, right? So the probability that one compound would continue, and because of lack of tox findings, was relatively low. So we thought, and our strategy for ASOs was, okay, try to use a CMS, so not to invest too much in the method development, and then maybe refine the assay and be ready for the clinic with something which is more suitable. But then we would invest more in the method development, in the capture detection probes and stuff.
Yeah, you make a really good point. The LBA methods or hybrid ELISA methods are going to take twice as long to kind of develop and validate as it would a mass spec method. That is a significant, you got to budget that in time-wise.
Yeah, from our experience, actually, the worst lead time is nothing to do with the lab. Is trying to get enough non-human primate CSF is ridiculous. Okay. And so depending on the sponsor as well, if there is so many CNS indication going on, right? So they always want to do CSF. So trying to get enough to do a full validation is problematic. It's about 150 mils of non-human primate CSF. It's not a good thing. And then some sponsors also are pretty insistent to use oncomouse as part of the validation, and you try to explain to them, hey, you can use QCs, right? No. I want oncomouse plasma. that is very hard to get, right? So a lot of times is these sort of things that it drives a timeline, and everything that you don't have lead time. No, our lead time is because we couldn't get all these matrices coming in. So that is actually, you don't think like that when you're doing small molecule because you never have that type of problems, right?
So, I mean, maybe just a last subject for us to cover before we wind this up. We'd mentioned a little bit before some regulatory aspects. Are there any particularly regulated feedback differences that you notice with oligos? And you're saying surrogate matrices, the regulators acceptable of surrogate matrices?
I mean, we have submitted surrogate. We have submitted oncomouse. I think it's depending on the sponsor needs. But I think there's definitely some matrices you need to be a little bit careful with that you need to use. But I think the regulations for the bioanalysis, we're using small molecule oligos, are very similar. The whole IND enabling package is very different, and you think a little bit differently. And then because your biodistribution you need to do very early on, and then in reality, as Iwa says, right, it's sort of something that is really interesting because you really want to understand the risk complex, the concentration. So you're now looking at PCR, whether you're doing DD or whatever you're looking at, right? But one of the questions that people is asking is, so now I fished out my oligos in the risk complex. Do I have metabolism in the risk complex? No, okay. Right? That is another question people is asking. Right. And that is a very difficult question to ask.
Yeah, I think when it comes to what we're just talking about here, anytime you can use a surrogate, if the guidelines are pretty clear. If there's you just can't get enough matrix, then you can use a surrogate. You just have to demonstrate it. But I do think regulators are more cognizant of some of the supply chain issues, and I think going back to COVID, that really exposed especially non-human primates. It would take a year to get it. It would be $250,000 or something ridiculous. So there have been a lot more publications and more submissions using surrogate, but I still think you might get some pushback. You got to be careful with doing that. I think if you can do it in the matrix, you should, obviously, but I do think there are more agencies are more amenable to showing in development, hey, this looks good, and then we're going to move forward with this surrogate matrix. Or maybe you create a surrogate for your standards and you just limit the QCs you're using as part of the true matrix. So there's some workarounds there. In terms of the regulations, I kind of think I touched on it a little bit earlier, but as we see more and more of these, I do think it's good to see a shift away from having to validate everything. It is traditional more to, hey, maybe validate CSF or validate plasma. And then tissues in and of themselves, in my opinion, are pretty difficult to validate. It doesn't matter if it's an oligo. Although oligos, we kind of touched on that a little bit, have a lot more intrinsic issues when you're looking at tissues. But from a regulatory perspective, I feel like it's kind of on par with others. But because of if you do have to move to some of these other platforms, I do feel like there's more leniency is the word that comes to mind for using surrogate matrix.
Well, personally, I don't see that to be an issue. And also, I mean, from a regulatory perspective, as long as we can justify, the proper use of the surrogate matrix. Tissues is a problem that indeed we have in the preclinical space. We don't typically collect tissues from the human beings, right? And there may be there may be exceptions, but biopsies, but not widely used. Tissue analysis also is something for which I'm not expecting, and we've never received any comments from the regulatory agencies. So we are not fully validating the assays for the tissues. The tissue, in my opinion, still remains a sort of semi-quantitative assessment because we can't determine the analyte recovery, right? So we typically adopt good assays, but the level of validation which is applied is definitely fit for purpose.
Yeah, but when we say the tissue validation, because we really cannot do the real matrix stability because the tissue need to be homogeneous. We cannot inject a compound in the tissue, then do that, right? So that's the one of the limitation. But compared to the other compounds, I think for the oligonucleotides, if it's a GCP, GLP type of the study, we still follow the same, regulatory. So SHM-10, for if we use the LBA assay, we follow the 20-25%. But if we use the LCMS assay, we follow 15-20. There was some kind of discussion about should we, expand the oligo LCMS acceptance criteria. But it is LCMS based, so we still prefer to use the 15-20. The struggling part is still the analog internal standard that really raise lots of challenges for the method. But our lab will still follow the small molecule LCMS approach. One of the things probably we can think about is the hybridized LCMS, what kind of technology, because the internal standard is added after the immunocapture, that step. So internal standard was not added at the beginning, so it cannot track from beginning to the end. Because LCMS assay, the criteria is 15-20. One of the main reasons it is because there is internal standard to track it down. So our method right now, still we can meet the 15-20 for the hybridized LCMS assay. But I have seen people, ask that type of question for that.
A question for you, because for hybrid LCMS, the internal standard can also be added at the very beginning, right? So it depends on the kind of internal standard, but if it is stabilized label, you can do with an analog for sure. But if you have a stabilized label, then it would follow absolutely the same fate as the analyte, and then you could add it at the beginning.
Lots of the vendor, they don't want to, because the cost itself for the stable label internal standard. We would love to have this type of, if it is, then yes, can hybridize at the beginning. But the method we have right now, the internal standards are added after the immunocapture step.
Yeah, I think 10 years ago, internal standard was an afterthought. Now, internal standard for hybridization is no longer afterthought. That's where the S34 comes in, thank goodness, because you don't need to worry about, you can get less than 10% now. Sometimes you get about 4%, 5% CVs, so that's important. I think one of the most important part I think we all need to remember is that the agency issued 2 guidance on oligonucleotide therapeutic development this year. Those are interesting read. It actually changes the whole way people do it. And then for last few years, they issued multiple guidance of oligotherapeutic nuclear development of N of 1, not N, no. So 1 patient, and those guidance are very interesting to read. And there are drugs out there going on. So oligos will never go away now. It's gonna stay.
This has been a really important and interesting conversation. I just want to thank you all for your fantastic contributions. It's been a real pleasure to have this conversation. So thank you to the audience as well for attending this Bioanalysis Zone panel discussion.